most of what I have to do here is now destructive testing. What I have to do is actually remove the sample and prepare it to be imaged. But it still gives us great 3D volume. So we'll stick it in a tube, we'll suspend it in a gel so it doesn't move because the scans take a long time. But again, we're going to rotate it over 360 degrees, capture thousands of images, and generate a 3D image. So this is what the flower looks like. I've been studying plants for over 30 years, and I've been looking at hundreds and hundreds of beautiful drawings and diagrams of what a plant looks like with the male parts, the female parts, the pollen up there, and the stigmatic surface, and the stigma, and the ovules on the inside. I've never understood what it looks like in 3D until December, when I looked at this, and suddenly realized what it looked like in 3D space. And purely from an imaging standpoint, this is tremendously valuable. Just to be able to see all of this in 3D at very high resolution. This is the other image from the other microscope. If we look at the exact same tissue, we can look at the whole thing at one resolution and then focus in on one region of interest, all just by controlling the microscope on a keyboard and a mouse. And I can even do it remotely from home. Team viewer is wonderful. But again, we have access to biological information now in 3D that has never been possible before, because in the past, the only way to look at this was you take that sample and then you embed it in plastic, or you embed it in wax. And then you take a knife and you cut hundreds and hundreds of physical sections, and then you look at one. And then you look at another one. You try to imagine what they look like in 3D. You just can't. You absolutely can't. So we have access to biology that it's never been possible before. But cool pictures, so what? Bring in knee to actually pull biology out of the scan. We can't do anything until it's actually identified, segmented, and measured. And that's where Knee and Mao just work their magic. I get some cool images, I get the fun part. They carry the weight of the lab by doing all this work. So we now, anything that's in a different color can be independently measured. So that's what we're doing with this kind of imaging data. We want to identify specific structures, we want to measure specific structures. Once they are identified on the computer, then it's literally just a couple of buttons, boom, measure whatever it is we're going to measure. We have pretty high resolutions. This is just one developing seed, and you can clearly see this is cellular resolution. This is just one slice, but we have that whole volume. Wow. We have the entire thing there. And again, you have one can get an appreciation for the biology in 3D that's just not possible anywhere else, any other way. There are microscopes that can do sectioning. There are laser microscopes, there are electron microscopes, but typically they can't look at something that big. And that is really the take-home message of the X-ray microscope. We're not going to go much higher in magnification. We're not going to go on a single cell and look at it, but we're going to look at that entire seed at cellular resolution, and that's where the power comes in. The other side of it is we're going to use this kind of technology in concert with the other imaging technologies and combine them all to look from very large to very small and have everything in between, all in 3D. But again, cool pictures, so what? Knee works for magic, and every different color is a different region, a different cell layer that we now can measure. Back to our roots. This is Nate's beautiful image of root system.